It is indeed my extreme pleasure to be able to introduce Dr. Hayden to our viewers here today. He is an accomplished scientist and physician, so I'm going to read his formal bio because I want you guys to hear just a small part of his numerous accomplishments that as someone in the Huntington's world for the last 25 years, I can tell you that Dr. Hayden is so well respected and inspirational in this community, and we have so many things as families to thank him for advancing the science of Huntington's disease. So that's the easy part, and I don't think I know anyone else as committed to finding a cure for families as Dr. Hayden. So he's the Killam Professor at the University of British Columbia and Senior Scientist at the Center for Molecular Medicine and Therapeutics. Dr. Hayden is also a Canadian Research Chair in Human Genetics and Molecular Medicine. Dr. Hayden was the President of the Global Research and Development and Chief Scientific Officer at Teva from 2012 to 2017. He led the approval of Austedo for chorea in HD, the second drug ever to be approved for HD in the United States. He's the author of approximately 900 peer-reviewed publications. He has focused his research primarily on Huntington's disease, translational medicine, including genetics, lipoprotein disorders, predictive personalized medicine, and drug development. Dr. Hayden also identified the first mutations underlying lipoprotein lipase deficiency and developed gene therapy approaches, resulting in the first approved gene therapy product, Glybera, in the world. Dr. Hayden is the recipient of numerous prestigious honors. Most recently, he was awarded the David Dubinsky Humanitarian Award from the American Friends of the Soroka Medical Center. He was inducted into the Canadian Medical Hall of Fame in 2017 and named one of PharmaVoice's 100 Most Inspiring People. He's been awarded an honorary Doctorate of Science by the Universities of Göttingen, you might have to repronounce that for us, and Alberta, the Luminary Award by the Personalized Medicine World Conference, the Diamond Jubilee Medal on behalf of Her Royal Highness Queen Elizabeth II, the Killam Prize by the Canadian Council of the Arts, and the Canadian Gairdner Reitman Award. Dr. Hayden has been awarded the Order of Canada and the Order of British Columbia. He was named Canada's Health Research of the Year by CIHR, which is the Canadian NIH, and he received the Prix Galien in 2007. Dr. Hayden is committed to empowering others. In addition to mentoring over 100 graduate students and postdocs, he's also a TED mentor. So we're so thrilled to have you, Dr. Hayden. I'm going to turn it over to you, and please feel free to correct any mispronunciations.
Well, that's embarrassing, you know, but thank you for that introduction. And what a privilege, and Terry, what a pleasure it is to be live with you here, and also to be part of this amazing organization, Help for HD, where the pioneers and founders continue to inspire the world and continue to bring their energy to bring light where there has been so much darkness, and how they've transformed their own personal darkness into light for themselves and shining light for so many others around the world. So it's a real pleasure to be here. Thank you.
Thank you. So we're going to launch into our 20 questions. And fortunately for us, for those of you guys who are here, Dr. Hayden is going to allow us to ask him a little bit about how he started his journey in our world here. So question number one, why have you spent your professional life working on Huntington's disease?
Well, you know, it's very strange. This was by chance. I was just finished medical school, doing a medical student internship in South Africa. And I went to a genetics clinic and started seeing families with what was not called Huntington disease at that stage. And I spoke to the physician in charge and I said, this looks like Huntington disease. And I was told that Huntington disease didn't exist, or had no prevalence in Africa, very little. And so I started following up with these families, just one family who had relatives all over. And as part of my PhD, I went to examine over 1,000 patients with Huntington disease, and in 11 institutions all over the country. And firstly, I didn't believe everything I was told. I would say, the lessons in South Africa have shaped my life, to think independently, to come to my own conclusions, to be data-driven, and then to be able to share or publish that. I was also spending a lot of time. In those days, you went to families' homes, and I went to homes. These were people mostly in the Cape Town area, disenfranchised by Huntington disease, but also disenfranchised by apartheid. And I went and sat with them. These were families often living in great poverty. But what moved me was their courage and their resilience and their warmth and the generosity of spirit that really touched me. And so I was imprinted early in my 20s to make a commitment to these families. Early on, we created the first association for families with Huntington disease in South Africa. And I'm proud to say we insisted that this be a multiracial organization right from the beginning. That was against the rules in South Africa. So one had to break some rules in an effort to really follow the principles that we so deeply believed in. I then was committed essentially to do whatever I could throughout my professional life to bring some alleviation, to fulfill the commitment I made to those families in South Africa, to see what could be done to try and alleviate suffering in multiple forms, defining the genetics, helping to understand where the gene was, doing first predictive testing, doing imaging, doing understanding mechanism, and then taking whatever we knew and committing that to try and translate that into services, not just products, but services and products that can help families. And that's what we've done, and that's what I continue to do. And every day, that is part of my own private mantra, to continue to think independently. I've taken people with me on the journey. Many of the leaders in Huntington disease today were trained in my lab, from Blair Levitt and Nando Skiteri and a whole host of others, and inspired a few people who themselves have now, that ripple of hope has spread to so many others who continue to make profound contributions. The founder of HD Buzz, Jeff Carroll, was a graduate student in my lab, and this story just keeps on going. So I'm thrilled to be part of this community, but we've still got a lot of work to do. There've been this year, there's been storms, disappointments in the area of Huntington disease, and I'm just pleased that we're not all in the same boat. I'm also one who believes there need to be multiple approaches to thinking about how we treat Huntington disease, not one mechanism, multiple mechanisms, combination therapies that in the end can do something to slow the progression and help patients with this disease.
Thank you so much. It sounds like that your early experiences in South Africa still serve as inspiration for you now.
I still think of those families. When I left South Africa, and by the way, the only reason I was able to get out of South Africa, because already I was in some difficulty not supporting the government, is that Marjorie Guthrie took me under her wing. And when I was in 1978, that's a long time ago, I went for my first time to America. And I met Marjorie at the International Congress for Huntington Disease. And I told Marjorie the story, and she said, "America needs you." And I said to her, "I won't come. I can't come unless you get me some landed immigrant status, some green card." She said, "I'm going to do that for you." And Marjorie ended up getting Senator Kennedy to write the letter to the INS saying I was an expert. And Kennedy had one condition. He said to me, "You have to go to Boston." And I said to him, Boston? Is Boston on the East or the West Coast? So I wasn't all that anyway, we went to Boston, and that's where we were. And Marjorie became a source of comfort, a source of support, and a beloved friend. So when I wrote the first author monograph on Huntington disease, which was published from Boston in the early 80s, I dedicated that to Marjorie, because without Marjorie, I would not have been in North America.
Well, thank you for answering question number 2. Just out of curiosity, I know Katie's probably going to get mad at me because I'm embellishing the story a little bit here. Did you ever meet Woody Guthrie?
No, because Woody Guthrie died in 1967.
Oh, sure, that's right. Yeah,
and I only came in the 1980s. But of course, I'm very close to Nora, and I know Arlo quite well. But Nora and I are dear and beloved friends. Nora's come to Vancouver to visit here. She's spoken to my students. I was there for the 50th anniversary of HDSA with Nora and the whole Huntington community in New York. Yeah, so yeah.
Yeah, I'm going back to script. Our next question is what was your involvement in getting approval for Austedo?
Well, when I went to Teva, one of the reasons is that you can't develop drugs in the academic community. You need the resources and the rigor of a company. Yeah. And so one of the conditions when I went to Teva was that I would be able to continue my work in Huntington disease. And we heard about this drug that had a mechanism that looked like it would help not slow progression, but rather have impact on some of the movements, chorea. And it worked in a very particular way. It reduced some dopamine levels from certain regions of the brain. And so I was able to find the company. This was a company called Auspex that had the drug in development, and I persuaded Teva to buy the company. This was billions of dollars, and we were able to bring that drug in and then complete the development and go to the FDA to get approval. And this became then the second drug ever to be approved. But that was just the first step. That was important and has been useful for families who have chorea. But my real commitment was also to do something, if I could, that could do something to patients' progression as measured by endpoints that the regulatory authorities would accept, such as total functional capacity. And that became the next task, which I can tell you about in a moment.
Right, thank you. So tell us what Pridopidine is.
So Pridopidine is that drug. So I brought that drug into Teva because I also had heard the voice of patients. And I've heard many people telling me that the drug didn't seem to have much impact on their career, but had impact on their progression in their own view. They were doing better. So we brought the drug in, and when we brought it in, it was still thought to be a dopamine modulator. And so we started a trial for movement. But soon thereafter, we recognized that we had the mechanism of action wrong. We were open to the new data, and the new data told us that this did not act on dopamine, but act on a pathway that was neuroprotective. In an effort to see whether this drug was neuroprotective, in Huntington disease, as everybody knows, you progress slowly. It is inexorable, but it's slow. So you need time if you want to see whether the drug has impact on progression as measured by some of those endpoints. You have to follow families and patients for a time, a minimum of a year. And we prolonged that study. We extended the study for a year. The total functional capacity, TFC, was pre-specified, and we just wanted to see whether there was any hint that this may have impact on TFC. And I would say, to our surprise and delight, and I'd say it's surprise, the drug had significant effects on TFC that stimulated us to then do a proper, formal, rigorous phase 3 trial in an effort to assess whether this drug could have impact on progression as measured by TFC. TFC measures the ability to do finances, the ability to continue working, the ability to take care of yourself, the ability to do household chores, the ability to live on without necessitating help getting dressed and other things. So it's it really is an index, and the FDA and the EMA, the European and American regulatory authorities, have accepted this as a single pivotal endpoint. When I left Teva, they allowed me to take the drug with me, and we had some people who'd seen some of the early data, and they were inspired by it. They recognized that while the drug didn't have that big effect on motor as measured by a total motor score, it appeared to have some impact on function and progression as measured by this TFC. And so we were able to go to the regulators, and they saw some of the data that we had seen, and they said, this is worth a trial that should be done. This was an uphill battle. We had to raise the money to do the trial. We had to get the regulators on side. But I must say, this has been an amazing experience. The families, this is an oral drug. That's very important because it can be taken easily, very accessible. It appears thus far to be safe and tolerable. And we were able to recruit 499 patients almost 2 months ahead of schedule during COVID. Everybody told us, you'll never be able to do this. And yet the families and patients responded with such positivity. In Canada, the United States, and 9 countries in Europe, all of them had to go through regulatory approval in each place. And the trial has been ongoing now for 15 months. It'll read out in the first part of next year, and we will learn whether we have an oral safe drug that appears to slow progression as measured by TFC or doesn't. And that's the purpose of a phase 3 trial. The FDA has seen now all the data, and they gave us what's called fast track approval. Fast track means they're going to walk in the journey with us. They're going to have this very close interaction. They're going to allow us to file things early. They're going to, and so this is, and they've accepted the endpoint and accepted the design. This community has been responding amazingly in participating in this. And this is an easy drug to take. And of course, I think about the families in South Africa that first inspired me. I do want to make sure that an oral drug like this is available globally with universal access, and hopefully in those countries that can't afford this at prices that are not going to make this out of reach for all families, certainly in the rest of the world. We're not there yet, but we have some hope on the horizon. And I think this is the early data made us realize it's not mission impossible to find some drug for Huntington disease. It's mission possible. And I'm one who believes you need multiple approaches to this. Huntington knockdown, Huntington lowering, this drug which acts on a particular receptor. You need multiple approaches because in the end, it's likely you're going to need combination therapy. Combination therapy that acts on different pathways in an effort to halt this really very, very tough disease. So we're in the middle of this. We're, I was part of the first people doing predictive testing. I heard what Carlos, and it takes a lot of courage. But of course, if there was a drug that had any impact on TFC, many patients and many people don't want to be tested because they say there's nothing we can do if we're carrying the mutation. If there was something that had some impact, there'd be many more people who'd get tested. It would be a whole boost for the whole community. And if we could find a way to have some impact, that would be really amazing. The company's tiny, but these people have been with me now, working with me for 8 years. They understand the mechanism. We've raised the support that we need. And so this is a journey that I've been on, initially from the mind, then to the heart, and then back to the mind, and combining all of that in an effort to do what we could do to have some impact. It's not a sure thing. There's no sure thing in drug development, but everything at the moment is looking good. We await the results with anticipation.
That's wonderful. Dr. Hayden, the next couple of questions had to do with how Pridopidine works and about the safety. And you did address that. Is there any other comment you'd like to make before we go on to the next question?
Well, it's sometimes hard to know how a drug works, but you know, we have ways in biology to answer that question. Because if you think it's acting on a gene, you can look at animals and you can delete one gene. And then, so for example, let's just say you look at normal animals and you give your drug and you see the effect and you see some effects as measured, and then you just delete one gene and you look, and if the effect is completely gone, then you know it's acting through that one gene. And so we know now, really over the last few years, how this drug works. It acts through a receptor called sigma-1. And sigma-1 is like a train hub that has impact on numerous pathways that are protected. It enhances the removal of toxic material. It enhances synapse function. It enhances connections in the brain. It increases factors like that are chemicals that are good for the brain. It decreases inflammation altogether, which acts in a neuroprotective fashion. Now we have to see whether this might work again in patients with early Huntington disease so that we can see the effect. But the mechanisms are quite clear. And those mechanisms are not just for Huntington disease. You know, the trial Mass General and Harvard Medical School have sponsored. We gave them access to the trial, and they're using this also in ALS, where some of those mechanisms of cell death are also apparent. And there are other mechanisms that may be used, other diseases where those mechanisms are conserved, causing injury, even though the disease is different. So there may be much more widespread application if successful in a few of the first ones.
So you mentioned that Pridopidine is being investigated in ALS. Any other disease entities that are giving it a try?
Well, there are, this mechanism is also been looked at in with other drugs that act on sigma-1. They're not quite as selective as this one. And by the way, it's just by chance. We didn't know, we had the drug, and then we saw how selective it was, acting on sigma-1 and not dopamine. We didn't know that. But there are other drugs that are less selective, but still act quite a big effect on sigma-1. They be using in Rett syndrome, in Fragile X syndrome, Parkinson's disease, and also cognitive impairment in Alzheimer's. So these are all the same mechanism being investigated in both neurodevelopmental and neurodegenerative conditions. We are the only people looking at Huntington disease, and right now the only ones in late-stage clinical development for ALS.
Yeah, good to know. So the next question is there a difference between past studies with Pridopidine and the current study?
Yes, big differences. So in the past, we thought this was a dopamine modulator. And the endpoint we had for that study was impact on motor function, because it was acting on dopamine. And the first trial, some early trials showed some success. Our phase 3 exploratory trial, phase 2, failed on that endpoint. It did not show significance. We now know why, because that's not the mechanism of action. So now we moved the total functional capacity, which was a exploratory endpoint in the prior study, we still had data, to now be the primary endpoint, because the biology has driven the clinical design, and the biology now has taught us what to look for the effect of this drug. So the trial is quite different. It's also longer. The initial trial was 6 months, extended to a year. This trial is 65 weeks. So why have we made it longer? Because if you want to look at a difference between a drug effect and placebo, the placebo should go down more. So you have more power, a bigger window to see whether your drugs have any effect on the slope seen in patients on placebo. So it's also being watched carefully for safety and tolerability. And so far, so good. It's looking very good. And the retention rate has been 97% after 15 months, which actually for any drug in any trial, you expect 10–15%. But we're seeing this will continue. There will be further dropout, but it's not going to be very substantial, and again talks to the safety and tolerability of the drug.
Yes.
That's for sure. We already are seeing that. Or even though we don't know who's on what, we just haven't seen anybody with many serious adverse events.
Yeah, that's so fantastic and actually so encouraging. You actually have shared this, but and why it's important to you, but let's just reiterate for the next question that how the drug is administered.
So the drug is given orally. It's given twice a day, once in the morning and once in the evening. It's a simple, there's no issues of drug supply. There's no issues of manufacturing and synthesis. It's easy to produce and it's easy to administer. It can be given with or without food, so there's no restrictions, but twice a day in a dose of 45 milligram tablet twice a day. Easy to take, not a big tablet, and easy to swallow, and so on. So safe and tolerable, and even in people with ALS, where there's often a lot of swelling of bulbar,
Yes,
had
yes.
no issues with the drug being administered.
That is so encouraging. You also told us this is being done in North America as well as Europe. Now that the study is fully enrolled, are there any thoughts to an open label extension?
Yeah, well, that's a great question, Terry. And of course, what's happening come April, there'll be the first patients finishing the trial. When they finish the trial, we won't know whether they've been on placebo or drug, because they finish the trial, and we'll only know that when all the people have finished the trial. But everybody who finishes the trial will then be able to go on open label for at least a year and probably longer. So they will now go on drug and continue to be monitored very carefully. This will give us more safety data. Now we know they're on the drug. And so everybody in the trial will stay on the drug at least for a year and perhaps for longer.
You know, that's a really interesting point, Dr. Hayden, because you're right, retention rates that high are really remarkable. And, you know, I think that speaks to several things, but I think also people knowing when they originally consent to come into a study, people knowing that they have that opportunity to actually get the drug makes the potential for that you might have in the beginning been on placebo more tolerable. It's kind of a I think it just gives them a gift, right, in terms of their initial participation.
Now, we've also allowed what's called expanded access. When the FDA gives you fast track, they also want you to make the drug available in a compassionate way. Yes. For people who, for whatever reason, couldn't get into the trial. Mm-hmm. So we have started an expanded access program, both for ALS, that Merrick Kutkovitz is running for ALS, as well as for Huntington disease. Both of them will have 24 patients, generally from countries where the drug is already gone through the regulatory process. So because then we just have to get permission to give this drug to others. We don't have to file a big regulatory file. So this would be available in United States, Canada, and 9 countries in Europe, including Italy, Germany, the United Kingdom, France, and others. And this is already underway. And we will do this to allow some people who, and the FDA asks for that, about giving patients access and in this way. So we have drug supply and we're working to do that. Just to say, we also, this trial is supported by the Huntington Study Group, which is HSG, who are helping to run the study, and of course, very expert in running trials in HD. And this has been, again, a very wonderful collaboration and cooperation in the United States and Canada, and then another CRO in Europe called TFS.
Yes, yes, yes. So this is an interesting question someone submitted, and you've kind of talked a little bit about this, but would you categorize with the signals you got with changing TFC, would you categorize this as a disease-modifying treatment or a symptom management treatment?
That's a great question. And, you know, I'm not categorical about this.
Yes.
What I'm categorical about is that this had impact on improving function and retaining function and delaying progression. If you want to say it's disease modifying, you really should do a different kind of trial, a trial where you also withdraw. So I don't like to use the word disease modification because it necessitates that when you withdraw the drug, patients do worse. I would say this is a drug useful for treatment of Huntington disease. It delays progression as measured by TFC if successful. And I'll let others argue whether we still don't know. Is it acting through symptom management? You know, that people are cognitive impact better, they're less depressed, but we do know from a preclinical phase that this is neuroprotective, that this protects neurons. It allows neurons to survive longer. You can transform a neuron in the striatum to a normal longevity even if they have the mutation in the presence of this drug. So it makes them more viable, less susceptible to death. Now, is that disease modifying or symptomatic? I would say we have some evidence for neuroprotection, and we're able to delay progression as measured by TFC. I'm not going to use the word disease modification because our trial wasn't directed to that. We're looking for something that can do something for patients and families around the issues that they worry most about. They can't walk, they can't work, they can't hug their children in the same way. They feel unstable. The disease is progressing by all measures. If we can slow that, that's enough for me. And I'll leave it to the other experts to define exactly the mechanisms. And that's still important, but we just want to have some impact on progression as measured by TFC.
You know, I totally love that answer, and I'm pretty sure everybody who's watching and or listening loves that answer as well. Thank you for being so clear about it. You touched a little bit about this, but if you want to make any clarifying comments on what does it mean to be fast-tracked by the FDA?
So fast track is it's not given to many, but the FDA has to agree with your design and your endpoints, the clinical design of the trial. So that's first. The FDA has to believe that your endpoint is meaningful and has the potential to really provide meaningful information. And the moment you have to submit a long and formal application to show why you believe. The FDA either accepts it or rejects it, and so not a very, it's a small proportion of those who get fast track. And then it allows you to have the FDA agrees that you can meet with them. They become partners in a totally different way in your phase 3 trial. Mm-hmm. Also, if you do have successful results, it allows you to be considered for priority review. It means that the review can go faster. It also allows you to file your new drug application in stages. So we could even file an NDA now, but without the data from the trial. But we do have some data on safety, clinical pharmacology, manufacturing, toxicity, and that's not going to change in the next year. We have all that, but it allows you to file it, you get some feedback. So it's a much more iterative, consultative, collaborative, cooperative process, and the FDA works with you on that. So we're looking to have meetings with the FDA coming up in the next quarter, where we discuss all of these issues and develop a plan with them. So they become your partners in helping to facilitate the trial, and we become very clear about what they need from us.
And I have to say, I think, on the other end of conducting clinical trials, I think that's so important and such a good tool in the toolbox, because we certainly know families struggle with how long it takes us to complete trials. They struggle with all the regulatory things that we have to face. And of course, those of us conducting them, we kind of get it. So I really appreciate you spending a little more time helping our community understand that, because it is really helpful in terms of having that designation as a clinical trial goes through. So our next question, and this may be a little challenging to describe too, but would this therapy be better for use in prodromal, early disease, or manifest and beyond manifest, or end stage?
Well, our trial is being done in early HD. That's stage 1 and stage 2, a TFC of 7 to 13.
Mhm.
Now, of course, if you have a neuroprotective agent and you want to protect neurons, you want to do that before you've had too much deterioration. We cannot resurrect neurons. So early would be better. We also know in prodromal, or people destined to be ill but not yet ill, they already have, there's a lot of functional reserve in the brain, and people don't manifest until it's sort of like drops and drops, and then finally it spills over into the clinical manifestations. So it is possible, if we were successful, that the drug would be good in early HD, but then you'd have to do the trial on people destined to be ill but not yet ill to show that you can delay the onset. That would be an important trial, but it'd be one you do when you know you can at least treat early HD. Now, to do a trial in late HD, it doesn't mean it's not useful, but the TFC goes down much less in late HD because, as you know, people can be not working, not managing their finances, not able to take care of themselves, have care at home, but they can still live for 10 or 15 years. The TFC at that stage is already very low. You've lost so much function, but so the change in late HD is much less. In early HD, about 0.8 to 1 a year. In late HD, 0.1 to 0.3. So if you want to know if it treats late HD, you'll have to do a much longer trial with more people to detect the difference, because the window to detection is much smaller. So we're looking to start off with early, where that's the bigger, obviously more to protect. And then we'd be very interested in prodromal and also late HD. And we'd be looking for an indication that says treatment of HD, because we still don't know how early you can go and so on. But certainly what makes it easier for early HD and for prodromal, it's an oral drug that has, at the moment, a very placebo-like safety profile. That makes, we have spoken about this like a statin. This is something you initially treated patients with coronary disease, then was taken earlier to prevent that. Maybe, we do not know, we'd have to do the trial to prove it, that we can delay onset in a carefully selected group of patients who are destined to be ill in a period that's soon, or some biomarkers, imaging or blood. There are some biomarkers you can use and see if you can slow the progression to onset. That becomes part of the work that follows this trial if successful.
Our next question, where would you recommend that our community go to find out more information?
Well, I would go to Prilenia.com. By the way, Prilenia, P-R-I, it's Pridopidine, and lenia is from the Greek, which means to soothe or to provide relief. So this is our hope and aspiration and prayer that this drug or something acts like it will be able to provide some comfort, some relief from the terrible suffering that families have. If you go to Prilenia.com, there are all the publications, and there've been many, all the details around the drug. Also on expanded access is there. This has to be, for expanded access, it has to be put forward by your physician, who will then manage you as part of that process. But it's all there, and I would say just to read, there's a good start. And if people want more of the science, there's a lot of publications on that website.
Perfect. So this is a crystal ball question. You have your crystal ball with you. How long will it take for this drug to be available for patient use if it's approved?
Well, it's a crystal ball, but it's actually, we know a lot already. So the trial, let's just say the trial ends sometime in the first half of 23. We will have the results a month or two later, very quickly. Let's say we're unsuccessful, the drug will never be approved. Let's say we are successful, and let's hope and pray for that. Then you would go into, we'd have to prepare our application, but a lot of our application will already be done because we'll have saved all the time. And we know for patients, if you're protecting neurons, every day counts. So you want to get in as quickly as possible. So we would hope to submit an NDA within some months. Depending on the FDA, you could have approval for distribution by sometime in the first half of 24. So that's just about 2 years from now. So that's not that far when you think about it. And again, we'll be filing simultaneously in the US, Canada, and Europe in an effort to get, but the US will be the first place where this becomes available. And let me just say, if successful, we do a phase 3 because we don't know, we have to prove to the regulators and to ourselves. I'm not interested in a drug that doesn't have an effect that's predictable. I want to make sure there's no harm to patients. Yes. That's very important. And right now, from the safety and tolerability, there appears to be no harm, and everybody appears to be doing quite well. So that's that, but we don't know the actual data that predicts whether this is an approvable drug or not. But we'll know this sometime about a year from now.
Perfect. Our last question, Dr. Hayden, is if it's approved, how long will patients need to remain on the drug?
That's a good question, but I would say this is going to be a drug that's going to be taken for a long period. Yes. Because if you want to protect your neurons and have some shield against further damage, or at least some mitigation of that damage, you probably need to take this, and it could be for many, many years. So that's also why safety and tolerability is so important. And ease of administration becomes very useful because people don't have to go to the clinic for the rest of their lives. They can take this at home. And it's also a drug that could be given by family physicians or general neurologists because it doesn't need specific procedures with which to give the drug. So this becomes much easier and accessible if the drug is successful in its results.
You know what? I'm going to get in a little bit of trouble here because I'm going to ask one question that because I know we're like, oh, but we're going to guess our time frame. But I have to ask: is it your anticipation that this drug would have to go through a specialty pharmacy?
No, I don't think so, because this is not going to, because this is a formulation's fairly straightforward. Yes. It's the other point about this is it's stable. The drug doesn't have to be kept in certain conditions of temperature. It's a very stable drug. We've had stability now that we've been able to continue to test. It's remarkably stable. And so I think it's going to be generally available if successful.
That's perfect. Dr. Hayden, I noticed that Katie and Katrina came on. We cannot thank you enough for coming on today, taking time, out of your Saturday, so that people in our community get firsthand information for this remarkable work. There is so much enthusiasm for this study, and you've just been wonderful in giving us clarity and motivation. And you have an army of people cheering you on and hoping for the best that comes out of this study. Thank you so much.
And thank you to Kate, the whole organization. This Helpful HD there was a real opportunity and a vacuum in this kind of interaction. And I've been so moved by people leading this organization, Kathy and others, and their commitment to do something for their children, grandchildren, for the future. And if we can just all work together, there's no such thing as you and I. It's us. We're only us. We're inextricably bound to each other in an effort to move this forward. And so for me, it's just a deep privilege. And I look forward to, and I'll just say one thing. I've learned so much from Huntington families. I was involved clinically, and I've just got a poem in front of me that this one person always used to say to me, "Hey, doc, you're working too hard. You need to," and this was a person who loved Tim McGraw. And she used to say, "I'm living like I'm dying, but you're living like you're going to live forever. You need to take some time out, spend time with your family, So what I'm saying is we also learn so much from each other. This is just not one way. I've shared some information today. but I'm very grateful for what I learn about resilience and courage and dealing with dark times and how people have coped with that. This is also very deeply moving personally for me. So it's a two-way street.
Thank you. Thank you, Dr. Hayden, so much for coming on with us today.